The ecological matrix of equilibrium. Within the broader planetary biosphere, natural ecosystems maintain this host pathogen equilibrium through dense, feedback-driven population mechanics that prevent any single species from achieving unchecked runaway domination. This is governed primarily by density-dependent selection filters. When a host population experiences an artificial surge in numbers and outgrows its ecological niche, the absolute number of susceptible individuals escalates, directly driving up the basic reproduction number of its associated pathogens. This spike in pathogen transmission triggers a localized epidemic, thinning the host population back down to its sustainable carrying capacity and preventing the degradation of surrounding ecosystems. Furthermore, pathogens function as primary balancing agents in biodiversity through negative frequency-dependent selection, a process colloquially understood as the maintenance of genetic polymorphism. Pathogens naturally and disproportionately target the most common, high-frequency genetic strains within a host population because those strains present the largest, most easily transmissible biological target. As the dominant host genotype is targeted and suppressed by the adaptive pathogen, rare genetic variants that possess alternative immune configurations are granted a competitive reprieve, allowing them to survive and propagate. This constant ecological sieve prevents the emergence of genetic monocultures, ensuring that natural biological conflict preserves the overarching genetic diversity and structural resilience of the global biosphere. Structural and cellular barriers. Before an invading pathogen can establish a metabolic foothold and initiate replication, it must breach the contiguous physical and chemical boundaries, separating an organism's internal milieu from the external environment. In multicellular lineages, this primary line of defense is anchored by the integumentary shield, which manifests as a specialized epithelium, such as the cornified epidermis in mammals, chitinous cuticles in arthropods, or lignified bark and flora. In mammalian systems, the stratified squamous epithelium of the outer epidermis undergoes continuous desquamation, a programmed shedding of dead keratinocytes that mechanically expels attached microbial colonies before they can penetrate deeper vascularized tissues. For internal tracts exposed to environmental influx, specifically the respiratory, gastrointestinal, and urogenital pathways, the host deploys a dynamic mucosal barrier paired with a kinetic ciliary escalator. Specialized goblet cells secrete a highly viscoelastic mucus gel composed of heavily glycosylated mucin protein, which functions as a molecular sieve to trap airborne and ingested pathogens. Within the respiratory tree, millions of synchronized, beating-silated epithelial cells provide directional kinetic force, driving this pathogen-laden mucus upward and outward toward the pharynx for gastrointestinal degradation or expectoration. This mechanical containment system is reinforced by a specialized suite of localized chemical deterrents secreted directly by epithelial tissues. In oral and ocular secretion, the enzyme lysosine acts as a primary biochemical barrier, systematically hydrolyzing the glycoside bonds between N-acetylmuramic acid and N-acetylglucosamine. In the peptidoglycan cell walls of gram-positive bacteria, inducing osmotic lysis. Pathogens that survive oral passage are funneled into the hyperacidic lumen of the stomach, where a profound proton concentration maintains a low pH unit. This intense acidity instantly denatures foreign microbial proteins, disrupting their tertiary structures and rendering their metabolic enzymes non-functional. Simultaneously, epithelial surfaces express an array of structurally conserved antimicrobial peptides, such as defensins and cathelicidins. These small, amphipathic, and highly catatonic molecules are electrostatically drawn to be negatively charged phospholipids and lipopolysaccharides, characterizing bacterial and fungal cell membranes. Upon contact, AMPs insert themselves into the lipid bilayer, aggregating to form lethal transmembrane pores that disrupt chemical gradients and cause immediate cellular lysis. Immunological defense systems. When a pathogen successfully bypasses these physical outer perimeters, the host coordinates an integrated, two-tiered immunological response designed to isolate, identify, and neutralize the threat. The innate immune response. The initial phase is governed by the evolutionary ancient innate immune response, which operates with immediate, nonspecific kinetics. This system relies on specialized tissue-resident sentinel walls, predominantly macrophages, dendritic cells, and neutrophils that express hard-coded pattern recognition receptors, PRRs, such as toll-like receptors, TLRs. These receptors recognize highly conserved immutable molecular motifs unique to microbial lineages, classified as pathogen-associated molecular patterns. Upon binding to PAMP, such as bacterial lipopolysaccharides or viral double-stranded RNA, these phagocytic cells initiate phagocytosis, engulfing the invader into an internal vesicle called a phagosome. This structure undergoes rapid maturation, fusing with the intracellular lysosomal sacs to form a phagolysosome inside the specialized vacuole. The pathogen is subjected to an array of hydrolytic enzymes and an intense respiratory burst, where NADPH oxidase generates reactive oxygen species, including superoxide radicals and hydrogen peroxide, to destroy the microbe's structural and genetic components. Concurrently, the innate arm activates the complement cascade, a circulating network of liver-derived plasma proteins. Upon detecting microbial surface anomalies, these proteins undergo a sequential proteolytic cascade that culminates in two distinct defensive outcomes: the chemical tagging of the pathogen for enhanced phagocytic clearance, opsonization, and the direct assembly of the membrane attack complex, which drills macropores into the microbial membrane to induce immediate osmotic collapse. The adaptive immune response. Because pathogens evolve too rapidly for hosts to rely exclusively on fixed germline-encoded receptors, jawed vertebrates develop the highly adaptive immune response. This secondary system operates with a multi-day kinetic delay but provides exquisite molecule-specific precision. The foundational engine of this system is somatic VDJ recombination, a process where developing B and T lymphocytes randomly slice and rearrange their own genomic DNA within a single lifetime. This localized genetic shuffling generates a nearly infinite repertoire of unique antigen receptors, allowing the host to match the hypervelocity of microbial evolution and internal somatic diversification. When an intracellular pathogen, such as a virus, successfully establishes an internal assembly line, cytotoxic T lymphocytes scan surface-displayed major histone compatibility complex class I molecules to detect foreign peptide fragments. Upon recognizing an infected host cell, the cytotoxic T cell releases perforin monomers to permeabilize the target membrane, alongside granzymes that activate the host cell's internal caspase cascade, forcing it to undergo apoptosis, programmed cellular suicide, to halt viral replication. Meanwhile, activated B cells undergo affinity maturation via somatic hypermutation inside lymphoid germinal centers, transforming into hyperspecialized antibody factories. These secreted immunoglobulins bind specifically to the surface antigens of extracellular pathogens, neutralizing their infectivity and targeting them for clearance. Following pathogen eradication, a selected subpopulation of these antigen-experienced B and T cells transitioned into a long-lived quiescent state, forming an immunological memory pool. This structural archive ensures that any secondary exposure to the exact same pathogen is neutralized instantly by an explosive, high-affinity immune response before clinical pathology can manifest. Behavioral immunology. Because the activation of systemic immunological defenses demands an immense metabolic sacrifice and can inflict severe bystander damage on host tissues through inflammatory pathology, natural selection engineered behavioral countermeasures to prevent infection from occurring in the first place. This suite of preemptive behaviors is classified as the behavioral immune system, an evolutionarily conserved avoidance program that acts as a psychological line of defense. In high-order animals, this system is driven by the primary emotion of disgust, an intuitive behavioral adaptation that functions as a motivational barrier. This sensory-evoked aversion program commands animals to instinctively avoid potent pathogen vectors, including fecal matter, decaying organic detritus, stagnant water sources, and visibly ectoparasite-laden or disease conspecifics. By transforming the detection of pathogen-rich environments into immediate avoidance behavior, the organism drastically reduces its probability of encountering high viral or bacterial loads, conserving its metabolic energy for growth and reproduction. For ectothermic taxa, such as lizards, amphibians, and insects, which lack the internal metabolic machinery to generate an endogenous pyrogenic fever, behavioral thermoregulation serves as the primary mechanism to combat systemic infection. When infected, these organisms actively seek out external thermal sources, such as sun-warmed rocks or microclimates with intense solar radiation, to raise their baseline body temperature several degrees above their normal physiological set point. This behavioral fever effectively slows down the replication kinetics of temperature-sensitive pathogens, allowing internal innate defenses to clear the infection. In highly integrated social and eusocial populations, this behavioral framework expands into collective defense strategies. Eusocial insects, including ants, Formicidae, and honeybees, Apidae engage in strict social distancing and proactive quarantine. Individuals that detect internal chemical cues of infection will actively self-isolate from the nest or be forcibly expelled by healthy workers to preserve the collection health of the colony. Among social primates, this dynamic manifests as reciprocal allogrooming, where individuals dedicated substantial daily time to inspecting and removing ectoparasites from one another's fur. This cooperative behavioral ritual directly mitigates group-wide disease transmission while strengthening social bonds. This dynamic provides a clear evolutionary precursor to the core premise of my unified hypothesis, that human collectivism and mutual aid are deep-seated, biologically hard-coded mechanisms designed to optimize group survival against external pressures. Part 2. The Human-Specific Anomaly. Section 1. Compressed chronology and temporal asymmetry. The human exploitation of pathogens represents a radical, non-linear break from the universal timeline of natural conflict. While nature operates on a deep time scale of billions of years, human biological weaponization has occurred in a hyper-compressed window of less than 4,000 years. This transition shifted biological conflict from a slow process of genetic adaptation to a rapid, technologically driven phenomena driven by conscious intent. The micro-scale chronology of human exploitation. The entire history of human biological warfare is compressed into an evolutionary microsecond, accelerating through 3 distinct technological phases that sharply diverge from the deep time mechanics of natural selection. While the natural pathogen arms race evolved over billions of years through non-teleological genetic trial and error, anthropocentric pathicide represents a rapid, intentional weaponization of biology. This abrupt transition marks a profound shift in the planetary biosphere where human consciousness bypassed the stabilizing feedback loops of the Red Queen hypothesis. By transforming pathogens from natural balancing agents of biodiversity into instruments of targeted demographic and political destruction, human exploitation detached itself from the ancient constraints of evolutionary equilibrium. Phase 1. Macrovectoring and passive dissemination. The initial phase of human biological exploitation relied entirely on macrovectoring, using naturally occurring epidemiological crises without any understanding of underlying microbial mechanics. Early human civilizations weaponized the natural decay of organic matter, transforming contaminated materials into primitive delivery systems. Ancient armies routinely hurled the cadavers of plague victims of decomposing livestock over the walls of besieged cities or deliberately fouled shared water sources to induce debilitating outbreaks of dysentery, cholera, or anthrax. This macrovectoring strategy reached a historic turning point during the 1346 Siege of Kaffa, when Mongol forces hurled plague-infected bodies into the Genoese trading posts, directly accelerating the spread of the second plague pandemic across Europe. These early biological tactics targeted the intersection of human immunity and societal infrastructure, forcing populations into high-density, low-nutrition conditions that artificially amplified the host density in the classical epidemiological equation. Phase 2. Laboratory isolation and aerosolized dissemination. The second phase emerged alongside the birth of modern microbiology and industrial manufacturing in the late 19th and mid-20th centuries. With the ability to isolate, culture, and stabilize specific pathogens in a laboratory setting, human warfare completely decoupled disease transmission from natural environmental vectors. State-sponsored programs systematically cultivated mass volumes of high-consequence pathogens, including anthrax, plague, and smallpox. Scientists resolved the natural decay of pathogens exposed to sunlight and open-air environments by engineering specialized liquid and powder stabilizers. This permitted the weaponization of diseases via fine particle aerosol clouds, bypassing the direct contact constraints that naturally keep high virulence pathogens in check. An accidental release of aerosolized anthrax spores from a military research facility in 1979 demonstrated the lethal efficiency of this phase by causing sudden, fatal inhalation anthrax in downwind populations, illustrating how industrial human intervention can bypass the natural respiratory mucosal barriers built over deep time. Phase 3. Genomic engineering and synthetic pathogenesis. The contemporary phase of biological exploitation is driven by genetic engineering, synthetic biology, and computational genomics. Rather than utilizing existing wild-type strains, modern biotechnology allows for the deliberate manipulation of microbial genomes, compressing millions of years of natural mutation into a single laboratory iteration. Researchers can alter pathogen genomes to artificially increase transmissibility, expand host tropism, or deliberately amplify virulence, bypassing the evolutionary fitness trade-offs that naturally penalize excessive lethality. By repurposing CRISPR-Cas9 systems, originally evolved as a bacterial defense mechanism against viral invasion, modern science uses these molecular scissors to insert custom genetic payloads into viruses and bacteria to render standard antibiotics, antivirals, and vaccines completely obsolete. Furthermore, the de novo chemical synthesis of viral genomes from digital sequence data eliminates the need to harvest physical samples from natural reservoirs, introducing unprecedented risks of engineered stealth pathogens designed to evade innate pattern recognition receptors or selectively trigger host-destructive cytokine storms. The evolutionary scale divergence. This macroscale acceleration of biological manipulation highlights humanity's stark departure from the natural checks and balances of the biosphere. While deep-time evolutionary warfare operates on a gradual scale of billions of years, driven by random mutation and limited by transmission virulence trade-offs, anthropocentric biological warfare operates on an immediate scale of under 4,000 years, driven by conscious design. This intentional manipulation decouples the pathogen from its natural requirement for host survival, replacing dynamic biospheric equilibrium with structural instability that threatens planetary carrying capacity. By compressing evolutionary kinetics into single generations, the anthropocentric anomaly threatens the systemic stability built over deep time. This dangerous divergence provides a clear historical bridge to the emergence of the resource-heavy Homo colossus, whose technological output disrupts Gaia's self-regulating equilibrium. The pre-microbial era, 1500 BCE to 1800 CE. For the first 3,400 years of recorded human conflict, the weaponization of pathicide operated entirely without an understanding of microscopic life, relying strictly on crude empiricism and visible cause and effect dynamics. The earliest documented instance of intentional biological warfare occurred during the Hittite plague, 1320 to 1310 BCE, when the Hittite Empire systematically drove tularemia infected rams and donkeys across enemy lines into the Arzawa kingdom. By exploiting the natural virulence of Francisella tularensis, They severely compromise the defensive capabilities of their adversaries. This rudimentary manipulation of natural pathogen reservoirs shifted into tactical infrastructure contamination during the medieval period, epitomized by the 1346 siege of Kaffa. Mongol forces catapulted the corpses of plague victims Yersinia pestis over the defensive walls of the Genovese trading post. This tactical maneuvering triggered a localized epidemic within the besieged populace, a catalytic event historically correlated with the introduction of the second plague pandemic into the European continent. By the 18th century, macro-vectoring tactics advanced towards exploiting environmental persistence. During Pontiac's War at the 1763 siege of Fort Pitt, British military officers deliberately distributed smallpox-contaminated blankets from a hospital quarantine to Native American delegations. This action weaponized the high durability of the variola virus outside a living host to devastate immunologically naive populations. The Microbial era, late 1800s to late 1900s. The birth of modern germ theory, pioneered by Robert Koch and Louis Pasteur, fundamentally transformed pathicide by compressing adaptation timelines from centuries to mere decades. By isolating the specific bacterial and viral agents responsible for infectious pathologies, human warfare advanced from passive environmental scavenging to industrialized cultivation. For the first time, state-sponsored laboratories could isolate, mass-produce, and stockpile monocultures of lethal pathogens, such as Bacillus anthracis, utilizing specialized synthetic growth mediums to generate highly concentrated biological payloads. This industrialization necessitated a parallel revolution in delivery mechanics to optimize dissemination. During World War II and the subsequent Cold War, massive national bioweapons programs, exemplified by Japan's Unit 731 and the extensive clandestine networks of the United States and the Soviet Union, shifted their focus toward aerosol weaponization. By milling pathogens into fine powders or liquid suspensions and pairing them with explosive or pneumatic delivery systems, military engineers learned to generate stable aerosol clouds. This strategic innovation bypassed the host's primary physical defenses, such as the integumentary shield, by ensuring that the engineered pathogens were directly inhaled into the respiratory tract. This approach completely uncoupled disease propagation from its natural, direct contact transmission constraints. The post-microbial and genetic era, late 1900s to present. The contemporary phase of biological exploitation relies on digital genomics, recombinant DNA technology, and synthetic biology, reducing evolutionary adaptation timelines from decades to mere weeks. The advent of gene splicing in the late 20th century, scientists gained the ability to directly manipulate microbial genomes, inserting targeted traits to purposefully bypass natural host defenses. Research can systematically engineer recombinant pathogens by splicing specific genes into benign or wild strains, granting them absolute resistance to standard antibiotics, introducing vaccine-evading surface proteins, or enhancing their structural stability to withstand environmental UV radiation. As the field transitioned into the 21st century, synthetic biology completely severed pathicide from the limitations of natural extraction. Utilizing computational platforms, researchers can now design novel or extinct viral configurations entirely from digital sequence data, printing live, functional pathogens de novo via automated chemical synthesis without ever needing an initial physical sample from a natural reservoir. This paradigm shift compresses millions of years of natural selection into targeted laboratory iterations, providing a potent tool for the runaway resource-hungry exploitation that characterizes Homo colossus. The kinetic shock of temporal asymmetry. The fundamental threat of human biological warfare resides in the severe kinetic mismatch it introduces between natural evolutionary defense systems and human technological velocity. In the natural order, a host population requires a generational filter spanning thousands of generations to register, select for, and stabilize genetic resistance alleles against a novel, high-consequence pathogen. Human weaponization instantly deploys optimized microbial threats, denying host populations the vast timescales required for natural selection to construct matching biological counter-defenses. Furthermore, anthropocentric pathicide completely decouples the pathogen from its natural host fitness cost. In natural systems, highly virulent pathogens are bound to a strict negative feedback loop. If a microbe replicates too aggressively and kills its host too rapidly, it burns out its own transmission pathway and faces extinction. Humans dismantled this systemic check and balance architecture by artificially preserving, culturing, and continually re-releasing the pathogen from state-managed laboratory reservoirs. This intervention renders the traditional transmission virulence trade-off model obsolete, allowing unchecked lethality to persist indefinitely. Finally, human delivery mechanisms are explicitly engineered to overwhelm the host's immunological infantry by altering the pathogen's natural site of entry. In natural, anthrax spores typically enter an animal host through superficial skin abrasions, resulting in cutaneous anthrax, a highly localized, slow-progressing infection that is easily identified and cleared by innate macrophages. In contrast, human weaponization technologically alters this dynamic by milling Bacillus anthracis spores down to a precise aerodynamic size of 1 to 5 microns. When disseminated as an aerosol, these microspores bypass the filtration architecture of the upper nasal passages and the ciliary escalator of the trachea, penetrating directly into the deep alveolar sacs of the lungs. This structural bypass allows the pathogen to be directly phagocytosed into the mediastinal lymph nodes, triggering rapid, hyperlethal systemic shock and septicemia that completely bypasses the tiered physical barriers built over deep time. Artificial selection as the functional mechanism of the human anomaly. Artificial selection represents the active operational mechanisms of the human-specific anomaly, systematically dismantling the multi-millennial timelines that govern natural evolutionary biology. In natural systems, pathogenetic lineages evolve through undirected genetic drift and complex environmental filters that favor long-term transmission and dynamic host-pathogen equilibrium. Humans completely bypass these regulatory cycles through deliberate isolation, selective cultivation, and the targeted genetic modification. By seizing control of selective pressures, human intervention converts wild, ecologically balanced pathogens into optimized, predictable weapons of mass lethality within single generation. This aggressive intervention funnels a diverse microbial population down to a single hyperdestructive trajectory, forcing traits that would be rapidly penalized and eliminated under natural conditions to proliferate unchecked. The mechanics of deliberate cultivation and selective breeding. Prior to the advent of modern genetic engineering, state-sponsored bioweapons programs utilized phenotypic screening and forced environmental pressures to accelerate the evolution of microbial weapons. Early 20th century biologists systematically harvested microbes exclusively from the most explosive, rapid, and fatal clinical cases, artificially prioritizing extreme virulence over transmission stability. To accelerate this pathogenicity, scientists utilized the process of serial in vivo passage, deliberately infecting a succession of laboratory animals. By repeatedly passing a virus of bacterium through a lineage of targeted hosts, such as primates or guinea pigs, the pathogen was forced to rapidly adapt to that specific species' immune architecture. This technique artificially compressed centuries of natural zoonotic adaptation and species jumping limitations into a matter of months. Concurrently, research subjected these isolated strains to environmental forces, growing pathogens under intense artificial conditions, including exposure to high heat, ultraviolet radiation, or specific chemical solvents. By harvesting the rare surviving microbes that withstood these extreme conditions, scientists created mutant lineages characterized by unnatural physical resilience. These engineered variants possessed altered spore walls or enhanced resistance to solar radiation, ensuring they could survive environmental degradation long enough to execute their strategic objectives. The genetic and synthetic disruption of evolutionary timelines. The advent of recombinant DNA technology and modern synthetic biology removed the reliance on random natural mutation, transforming biological modification from a game of chance into an industrialized engineering discipline. The transition erased generational scale adaptations timelines, allowing scientists to use molecular tools or directly manipulate pathogen genomes. Through the engineering of plasmid-mediated resistance, bioweapons programs historically introduced foreign rings of double-stranded DNA containing multiple drug resistance genes into standard pathogens like Yersinia pestis or Bacillus anthracis. This targeted modification renders standard civilian medical stockpiles, such as ciprofloxacin or doxycycline, instantly useless, effectively shattering the host's technologically acquired herd immunity. To complement this resistance, geneticists can execute deliberate antigenetic shifting, rewriting the genetic code for a virus's surface epitopes. By altering these outer molecular patterns, the pathogen becomes structurally invisible to the host's existing vaccine-induced or naturally acquired immunological memory, guaranteeing unmitigated cellular infiltration. Furthermore, gain-of-function research allowed for the functional optimization of pathogens to withstand anthropocentric delivery systems, because wild-type vegetative bacteria often dry out and die quickly. When aerosolized, researchers utilized genetic engineering to modify the cellular membrane structures of metabolic pathways of these organisms, allowing them to survive the extreme physical shear forces of explosive release or high-pressure aerosol dispersion nozzles. This structural optimization is frequently paired with toxin splicing, a process where regulatory genes that normally downregulate a pathogen's replication or toxin output to preserve host longevity are systematically knocked out. Conversely, foreign genes encoding hyperlethal proteins, such as introducing the botulinum neurotoxin gene into otherwise manageable intestinal bacteria, are inserted to maximize cell-level destruction and induce rapid systemic collapse. Ultimately, modern automated gene synthesizers allow for de novo synthetic assembly, completely bypassing the requirement for a natural microbial reservoir. Utilizing Utilizing digital DNA sequence data, researchers can construct infectious viral genomes from raw chemical building blocks without ever needing to harvest a live sample from nature. This approach removes the evolutionary baseline entirely, as extinct pathogens like the variola virus or completely novel, unevolved chimeric viruses can be generated into a laboratory over a few weeks. This timeline erasure leaves the modern human immune system completely unprepared, exposing it to synthetic threats that have no historical or evolutionary precedent. The destruction of the evolutionary fitness cost. As established in natural systems, adaptations that increase virulence typically carry a severe metabolic or transmission tax that causes the pathogen to self-limit and burn out in the wild. Artificial selection deliberately breaks this defensive feedback loop by decoupling pathogenicity from environmental survival constraints. Within a state-managed laboratory setting, a hyperlethal engineered pathogen is completely freed from the basic requirements of evolutionary fitness and independent survival. Humans provide optimal growth media, temperature-controlled bioreactors, and artificial chemical stabilizers, maintaining the pathogenetic lineage in a state of artificial life support, because human military systems handle the mechanics of transmission via aerosol generators, cluster munitions, or targeted contamination delivery systems. The pathogen is entirely released from the natural requirement of keeping its host immobile to facilitate spread. This intervention produces an asymmetric result where the pathogen can maximize its virulence to absolute lethality without experiencing any corresponding drop in its basic reproduction potential. This uncoupled biological force possesses no analogy in the history of natural evolution, representing the exact type of runaway, resource-heavy exploitation that characterizes Homo colossus and threatens the self-regulating stability of the Gaia organism. The conceptual divergence, coexistence versus elimination. Asymmetrical intent represents the philosophical and strategic core of the human-specific anomaly, engineering a direct inversion of the evolutionary mechanisms that govern the biosphere. While natural evolution operates under the strict kinetic constraints of the transmission virulence trade-off model, which forces pathogens to stabilize at a dynamic equilibrium that preserves the host reservoir, human weaponization pursues an entirely opposite trajectory. The objective of biological warfare is the deliberate design and execution of the systemic crisis. It seeks to maximize immediate lethality, fracture species barriers, and trigger widespread biological collapse to achieve absolute strategic domination. This baseline intent fundamentally conflicts with the evolutionary directives of nature, which lean toward host optimization and long-term ecological balance. In a natural system, an overly aggressive pathogen faces severe negative selection pressures, driving it to co-evolve alongside its host community and transforming acute, lethal infections into chronic, manageable conditions over deep time. Human weaponization intentionally reverses this process by selecting exclusively for acute, explosive, and unmanageable clinical outcomes designed to cause total system failure. Military and strategic planning values biological agents precisely because they do not behave like conventional kinetic weapons. They are selected for their capacity to introduce a state of asymmetric shock that simultaneously disables or destroys vast percentages of a target population. This tactical disruption is engineered to overwhelm medical infrastructure, collapse child governance, and paralyze economic networks, using the pathogen as an instrument of complete systemic attrition. Furthermore, while natural pathogens diversify to fill specific bounded ecological niches, human weaponization narrows its focus to destroy foundational infrastructure. This manifests not only as anti-personal agents targeted at human populations, but also as anti-crop and anti-livestock agents designed for agro-warfare. By engineering pathogens to decimate primary agricultural systems, human actors use artificial selection to trigger artificial famines, proving that anthropocentric exploitation targets the very network of life to assert dominance. The strategic matrix of artificial lethality. To achieve this asymmetrical intent, human designers prioritize and amplify specific pathogenic traits that natural selection actively works to suppress or eliminate. This strategic framework operates through a dual approach, maximizing case fatality rates while manipulating incubatory delay to bypass standard public health defense thresholds. Natural selection typically maintains pathogen case fatality rates within balanced boundaries, ensuring that a sufficient number of hosts survive to maintain transmission channels. Asymmetric weaponization explicitly seeks out or engineers agents characterized by near absolute lethality. Pathogens such as aerosolized bacillus anthracis, inhalation anthrax, or pneumonic Yersinia pestis are chosen because their unrelated, untreated mortality rates track between 80% and 100%, transforming a biological entity into an absolute terminal event. This lethal capacity is optimized through the deliberate manipulation of incubatory delay. In nature, highly lethal pathogens generally present immediate, severe symptoms that alert the host population, triggering behavioral isolation or social distancing that limits the outbreak spread. Human military designs counter this safety mechanism by favoring an asymmetrical, asymptomatic incubation window. A pathogen engineered to remain completely silent for days or weeks allows infected individuals to move freely through civilian and logistics networks. This dynamic maximizes the stealth dispersal of the agent across geographic boundaries before the first clinical case is detected, rendering early containment protocols obsolete. Ultimately, the strategic optimization aims to completely bypass the immunity threshold of the target population. Natural systems rely on herd immunity, where a percentage of resistant or exposed individuals forms an immunological shield around vulnerable members. Asymmetric weaponization dismantles this defense by deploying novel, exotic, or genetically modified agents to which the target population has zero baseline immunity or historical exposure. By introducing these unmapped pathogens, human intervention turns an entire community into a completely defenseless fuel source for an engineered epidemic, ensuring that the resulting crisis spreads unhindered by the host's evolutionary history. The ecological consequence: uncoupled destruction. By forcing a pathogen to operate with asymmetric intent, human intervention permanently severs the biological checks and balances that prevent natural ecosystems from collapsing. In the natural order, pathogens are bound by strict host specificity, preventing a single microbial mutation from wiping out entire multi-species webs. Human bioweapons programs historically sought to eliminate these specific barriers, deliberately broadening host ranges or weaponizing highly promiscuous zoonotic agents like Francisella tularensis, which can infect hundreds of diverse mammal species. This intentional expansion ensures that the engineered pathogen cascades across taxonomic lines, maximizing collateral environmental damage and destabilizing regional biodiversity. This destruction is magnified by the introduction of permanent environmental poisoning. In nature, a pathogen typically decays or enters a dormant state when its host density drops, preventing the permanent sterilization of an ecosystem. Human weaponization bypasses this environmental baseline by blending pathogens with specialized industrial additives, such as anti-agglomerants, microencapsulation polymers, and chemical stabilizers. These industrial coatings protect the microbes from ultraviolet radiation, desiccation, and temperature fluctuation, locking them into an undegradable, highly persistent environmental state. Spores, or engineered viral particles, are rendered capable of contaminating air, soil, and water systems for decades after their initial release, creating a long-term toxic footprint. Finally, this uncoupled destruction completely shatters density controls. While nature utilizes disease as a regulatory feedback loop to manage overpopulated species and maintain carrying capacity, human deployment forces an artificial epidemic curve onto low-density or highly stable populations. This bypasses natural ecological balances entirely, shifting the role of pathogens from stabilizing balancing agents to agents of extinction. This runaway pathicide provides the clear biophysical evidence of the resource-heavy Homo colossus footprint, demonstrating how human technological intervention overrides Gaia's self-regulating equilibrium and sets the stage for the deep-seated transgenerational trauma outlined in my unified hypothesis.